Hi, this is Tim Weber from South Texas College coming to you. Going to be talking today about how our biology is tied into our psychology. So let's bring that up on the screen here. And if you look in your textbook, you'll notice they've called this chapter the biology of mind. And what they're trying to point out is that these two things, the mind and our biology, are inextricably linked with one another. Most psychologists will say anything that is psychological is at some level also biological. And I mostly agree, although I'm open to the possibility maybe something psychological has no physiological correlate. But if that occurs, it's rare, if at all. Okay, so then having a look at the cartoon here, the leader is telling the students the body is made up of millions and millions of crumbs. Of course, we're not bread, so if they were speaking to us, this would be bodies made up of millions and millions of cells. And cells that take on great importance for us are neurons, specialized cells that carry information throughout the body. Neurons also are like mini decision-making devices, as we'll see. So important in our psychological processes. Neurons then exhibit a number of parts that you should be aware of, and that you'll understand things better once you know what these are. First of all, you have the cell body, that rounded area here, and this carries on activities that keeps the neuron alive and provides energy for its activities. You then have these bushy branching structures here. These are called dendrites. Dendrites then have the task of receiving information that is coming from other cells or from other neurons. Well, neurons are cells too, but okay. So incoming information is what dendrites are all about. Then we have this longer cable-like structure here called the axon. And this axon conducts messages away from that cell body bound to stimulate other neurons, muscles, or glands. And it does so as it carries a neural impulse, which is also known as action potential. This is an electrochemical process that moves chain-wise all the way down that axon until it reaches these terminal branches here at the end of this axon. This is where the axon then forms a connection with other cells. This is the junction between that neuron and another cell. Many neurons also have a myelin sheath, which then covers that axon. The myelin sheath has a number of functions. One is it insulates that axon. It keeps that neural impulse from going places where it shouldn't go, just like the rubber coating on the wires in your house keeps the electricity from not going places where it shouldn't go. It also enables that neural impulse to travel much more rapidly. It turns out that neurons that have this myelin sheath develop it as we mature as children, and that enables us then to process information more rapidly and also an overall greater amount of information processing. So that accounts for why the 13-year-old can process information more rapidly and more fully than, say, a 6-year-old. The next thing that we want to look at is just what causes this action potential to occur, what causes this chemical reaction that travels down that neuron. And for that, we're going to switch to a different screen here. Let me see if I can bring that up for you. Here we go. And let's just do a little bit of work on our whiteboard, we'll call it here. So first of all, let's make this axis of the diagram to be the electrical charge. Okay, and this will be going up. And then this axis will be time passing. Okay, so when a neuron is just sitting around and really not doing anything, it has a slightly negative electrical charge, something around minus 65 millivolts, if I remember correctly. And so there it is, just sitting there not doing anything. This is referred to as its resting potential, that slightly negative electrical charge. Why is it called that? Well, it's resting, it's not doing any work at that moment. Potential, it has the potential to do some work given the right conditions. So it's kind of like, well, maybe let's say if you have a car, the battery in your car is sitting there charged, and it has the capability to do some work if you engage in the right sequence of actions. Well, same thing here. It's ready to do some work. It's not doing anything, has the potential, has the charge to do so. So nothing happens until various forces cause that electrical charge to rise to about minus 55. And at this point, we say it has reached its threshold. And this causes openings within the cell membrane of that axon to open, allowing positively charged molecules to rush in to that axon. And so what happens when you add all kinds of positively charged molecules, the electrical charge is rapidly going to spike. And this spike then is what is referred to then as that action potential. In fact, the axon will actually depolarize. In other words, it will go from negative to a positive value, about plus 40, and then it reaches its peak. Once it reaches that peak, all the energy has been discharged, then this whole process travels down the axon, and as it does so, each place where it occurs, there then is our other channels within that axon that open up, allowing those positively charged molecules to be forced back out, kind of like if you blow up a balloon and the air wants to get back out. Well, different positively charged molecules are forced back out. So what happens then, the electrical charge drops back down, eventually reaching that resting potential again. Now, during the time that this electrical charge is returning to its resting potential, the neuron is unable to produce another action potential. So we call this time here, when it cannot fire again, the refractory period. It's kind of like a one-shot rifle where you have to reload it every time it fires before you can fire again. It has to recharge before it can fire again. Now, this whole process then will travel down that axon until it reaches those axon terminals we talked about earlier. Now, we should know that this is an all or none phenomenon. Okay, so either it fires one of these or it doesn't. Nothing happens till it reaches this threshold. You might think of that kind of, again, using the gun analogy. You can put your finger on the trigger of a gun and you can put a little pressure on it even, and nothing happens until what? You reach a certain amount of certain number of pounds of pressure, and then suddenly, boom, the trigger breaks and you have the thing fire. Same thing with the neuron action potential. Now, also in terms of intensity, this is also an all or none process. You don't have a weaker or stronger action potential. Again, just fire it like firing a gun. Doesn't matter how hard you squeeze the trigger, you always get the same intensity of the thing firing. Same thing here. Now, you might be looking at it and say, well, if that's the case, and neurons are involved with, say, our vision or hearing, well, how do we tell the difference between an intense stimulus, a loud noise, or a bright object versus, say, a dim one? Well, that has to do with either the number of times per second this whole process occurs, or the number of neurons that are activated by the stimulus. It depends on the sensory system. Okay, so I think we got most of the things you need to know there. You can back this up and re-diagram it for yourself and so forth as well if you need to. So let's go back to our original screen then. Here we go. As you can see here in this illustration, again, that whole process traveling down the axon, first charging positively and then discharging again. Okay. Oops. Every time I do this, I have to get my pointer back again. Okay. All right. So now eventually then this action potential arrives at the synapse. The synapse is the connection point between the sending neuron that is sending this message and some other cell that is receiving the message. The synapse then actually is a tiny gap between that neuron and that next cell. This synaptic gap or synaptic cleft then is a little bit of a problem for us. How does the message get across from the sending neuron to the receiving neuron? Well, what scientists learned is it does so through the mechanism of chemicals that travel across the fluid in this synaptic gap and eventually contact that receiving neuron. We call these chemicals neurotransmitters. These neurotransmitters then, chemicals being released from a sending neuron, and as they float through that synaptic gap, if they encounter a receptor site, which they fit in with, then they can bind to that receptor site and thereby influence that receiving neuron. Neurotransmitters are of two basic types. We have those that are excitatory, and these excite other neurons or other cells and make other neurons more likely to produce their own action potential. We also have other neurotransmitters that are inhibitory, and you guessed it, these make other neurons less likely to produce their action potential. You might say it sort of calms them down. These neurotransmitters then work in a lock and key fashion. Okay, so maybe you've lost your keys at home, and so you go to the drawer that has a bunch of keys at home. Almost everybody seems to have a drawer like that at home. And you get out all those different keys and then try to find one that will fit in that lock. Same thing here. That neurotransmitter goes bumping into that receiving neuron. And if it then bumps into a receptor site that matches with it, it can lock on and bind up. If it doesn't, nothing happens with it. Now, once these neurotransmitters are found in that synapse, in some cases, there may be an excess of that neurotransmitter. And so, what happens to that neurotransmitter? Of course, the first thing is yes, it might bind up with additional receptor sites on that postsynaptic neuron, postsynaptic being the one that is actually receiving the information. On the other hand, if it's just left out here, the process of reuptake may occur. That neurotransmitter may be reabsorbed by that presynaptic neuron and stored in these little vesicles for later use, recycling. On the other hand, that neurotransmitter left in this synapse may be broken down by enzymes, chemical substances that can break it down into substances that have no effect. So enzyme deactivation, disassembling that neurotransmitter. Or that neurotransmitter might bind up with autoreceptors that are found on the presynaptic sending neuron. So autoreceptors, in this case, if it binds here, this is going to send a signal to that neuron that there's plenty of that neurotransmitter already in the synapse, don't need to send any more out there. So when it binds with an autoreceptor, it downregulates the amount of neurotransmitter being released. And then finally, that neurotransmitter left here might just drift off to parts otherwise. Notice that all of these mechanisms, whether it be reuptake, enzyme deactivation, binding to autoreceptors, prevents that neurotransmitter from constantly overstimulating another neuron. So it keeps things within homeostasis, within the right range. So let's have a look at then how neurotransmitters have their influence. So one neurotransmitter named serotonin has been indicated in helping to regulate our moods, to keep us from getting too sad and that sort of thing. You can see various areas of the brain that are primarily circuits that are activated by serotonin. And so if there is a lack of serotonin, in some cases, this may produce depression. Or if it is lacking, it is associated with depression. Now, another neurotransmitter, dopamine, if we have an excess amount of dopamine, then we get schizophrenia-like symptoms. Schizophrenia being what? A very serious psychological disorder. Just to give you some ideas of some of the possible symptoms of schizophrenia, could be hallucinations, paranoia, delusions, false beliefs and really not supported by reality. The person might withdraw or might engage in repetitive behavior. They might become frozen and catatonic, just all kinds of things, inappropriate, bizarre ideas, and you name it. Yeah, pretty significant. So more is not always better is the thing you should take away from that. On the other hand, when there is a deficiency of dopamine, then we get Parkinson's-like symptoms. Parkinson's being a movement disorder that starts out with tremors and eventually the person is unable to control their movements and also ends up influencing some cognitive functions as well. So once again, that side of things is not good either. A happy medium is important with many neurotransmitters. So here's a list of neurotransmitters we've actually, scientists have actually identified well over 400 of these things, but here are some of them that are most critical to our psychological functioning. We'll just go down the list. We have acetylcholine, or acetylcholine if you're British, I guess. Um, Notice can be abbreviated ACH. And this is the one that, when released, stimulates muscle cells to contract within our body, skeletal muscle. Also is involved with a certain type of learning, the type of learning that occurs when we associate one event with an event that occurred consistently immediately after. And acetylcholine also important for memory functioning. Interestingly, when people develop Alzheimer's disease, neurons producing acetylcholine deteriorate, and so does their memory functioning. Next one, dopamine. We talked about that in terms of importance for movement and importance for normal perceptual functioning. Notice it has a number of other functions as well. Important for a different type of learning, something we call operant conditioning. This is the type of learning that occurs when we do something and we experience a consequence of that behavior. If that consequence was desirable, we'll tend to repeat it as a result. We've learned that it produces a desirable consequence. That's a type of learning we need dopamine for. Also is important for emotions, and in particular, emotions that have to do with curiosity and being motivated to pursue a goal. Each time we move closer to some goal that we have and move towards it, we get a release of dopamine. That's rewarding, so we continue on motivated to move towards that goal. Okay, serotonin, we already mentioned how that's involved with regulating our moods, also involved with appetite, hunger, sleep, and arousal. Now, when we talk about arousal in this class, it's not always sexual, but it could be simply arousal in terms of just how physically and mentally activated we are overall. So notice that undersupply of that linked to depression, also linked to problems like what? Appetite, eating disorders, sleep disorders, insomnia, as well as lack of energy, lack of arousal, can't get going, that sort of thing. Okay, so bunch of things. And if you notice, wow, a lot of those tend to go together. Think about it. When people get depressed, often their appetite changes. They may lose their appetite. They may have trouble with their sleeping patterns, and their arousal fails. They just feel like they have no energy, fatigued all the time, things like that. Okay, so kind of all makes sense. We also have then norepinephrine. Now, this one is closely related to epinephrine. Does that help you? Maybe not. But what if I told you, well, the other fancy name for epinephrine is adrenaline? And you say, oh, everybody knows what adrenaline is, right? It gets you all worked up and excited and all of that sort of thing. Well, norepinephrine has similar effects. Notice increases our alertness as well as our arousal. Again, it prepares us to be active and makes us feel like being active. Notice a lack of norepinephrine also can depress mood as well. Then we come to GABA, and don't worry, you don't have to memorize the long form. If you know GABA for our class, that's good enough. This is, it says, a major inhibitory neurotransmitter. So what is this going to do in terms of our nervous system? It's going to calm the nervous system. And indeed, GABA is related to the management of anxiety. When there's an undersupply, people may experience seizures, tremors, shaking, insomnia as well. So, pretty important one. Then we have glutamate. Glutamate, very prevalent, is a major excitatory neurotransmitter. So what's it do? It gets our nervous system cranked up and activates our nervous system. Notice though, an oversupply can overstimulate the brain, produce migraines or seizures, even kill brain cells. Notice also that this may account for why some people have trouble if they ingest too much MSG, monosodium glutamate. This is a substance that may be added to food. They often call it a flavor enhancer, but the downside is that for some people, if they're sensitive to it, they can experience some of those effects. Then we've also added to our list the endorphins, which is actually a whole series of neurotransmitters. What these have in common is these are capable of dulling pain and producing a sense of calmness, a sense of wellness, a feel-good sort of situation. Okay, I know textbook says something about, I don't know, pain and pleasure, but that's just too vague. So think about dulling pain, producing calmness. Now, when are endorphins most commonly released in massive quantities? Well, there's a couple things that will bring on the release. One of these would be a very serious physical injury. And you might have observed this, people having received a serious injury may be sort of oblivious to it. They're so common, you think, oh my gosh, why aren't they freaking out? And whereas you might think they should be screaming in pain, they may be just trying to go on about their business. We see that sometimes in accidents. Or had a friend of mine broke their foot in the first quarter of a football game and played the rest of the game. It wasn't the adrenaline, which many people think, maybe that helped a little bit and kept them activated. But the fact is that massive release of endorphins meant he didn't feel the pain as he probably should have. Now, a second thing that will cause a very significant release of endorphins is vigorous exercise, particularly if it's performed 20 minutes or more. So that might account for why people, after they exercise, they say, oh, I feel so much calmer after I work out. And those aches and pains that I got, after I work out, I feel better. They don't notice them so much after the workout. That's probably why. Okay, so pretty interesting substances. Let's go on. All right, so some substances then are similar enough to a particular neurotransmitter that they can actually bind up with that neurotransmitter's receptor sites. So when this happens, that chemical substance can produce the same effect that that neurotransmitter does. In this case, we call that an agonist for that neurotransmitter. An example of that would be morphine. Morphine can bind up with endorphin receptors. And what is the effect of morphine on people? It relieves pain and they feel more calm. That's one of the best painkillers we have. Although, we have to be careful not to abuse it, because if we abuse it, then our body stops making endorphins. And then if we quit the medicine, then we have neither the morphine nor do we have the endorphins, and we feel really rotten. But at any rate, notice this is how an agonist works. Other substances are similar enough to a neurotransmitter that they can bind up with their receptor site, but not similar enough to have the same action. It's almost like when you're looking for that lost key through all those keys in that drawer, and you finally find one that goes in the lock, but then you find out it goes in the lock, but it doesn't turn lock. Yeah, that's the situation. So notice these then actually block that neurotransmitter from binding up with the receptor sites. They occupy those receptor sites and prevent the action of that neurotransmitter. So a good example of that is the substance karate. And this is a poison that indigenous groups have used for thousands of years. They'll put it on their arrows or their spears, and then they go to hunt for their dinner. And as they hit an animal with that, it goes in the animal's bloodstream. And what happens? The karate will bind up with acetylcholine receptors in the animal's body, and therefore, what? The animal cannot contract the muscles. Okay, and so the animal becomes paralyzed. They go pick up the thing and go have lunch. So antagonist blocks the action of neurotransmitter. Okay, so that's enough for neurotransmitters. Now let's have a look at something else. So have a look at the cartoon. So our subject in the cartoon is saying, you're certainly a lot less fun since the operation. Well, what has happened to her friend? Notice there's really nothing left but just a brain. Yeah. Now, we kind of tend to equate our brain with our existence and who we are as a person. But you have to admit that if we were only a brain, we'd be in a pretty tough situation. The brain has no receptors that can detect what's going on outside of itself. The brain has no ability to move and interact with the environment on its own. It needs our body to do these things. And so, yeah, if you're without your body, you're in pretty much a deficit state. So how does our brain then accomplish these tasks? How does information from our eyes and ears actually get to our brain to inform it what's going on? How does our brain activate the muscles of our body in order to move around and accomplish tasks? Well, the answer, of course, is it does through the nervous system. So the nervous system forms a link between the brain and body. Here you see how the nervous system can be broken down into a number of subsystems. It can, first of all, be broken down into the peripheral and central nervous system. The central nervous system being made up of simply the brain and spinal cord, the part you see in green here. The peripheral nervous system then is broken down into autonomic and somatic systems. The somatic peripheral system controls voluntary movements of our skeletal muscles. It also, by the way, carries information from our body, touch and hot and cold, pain and so forth, also from the body as well, back to the central nervous system. The other division of the peripheral nervous system is called the autonomic nervous system. Notice it sounds a lot like automatic, and in fact it sort of is automatic because it controls the actions of internal organs and glands without any input, without any conscious effort on our part. It does so automatically. So what kinds of things? Regulates our digestion, for example, regulates the tiny muscles in our blood vessels that regulate our blood pressure, regulates our breathing and heartbeat. With one interesting exception, breathing is about the only thing that is under both autonomic as well as somatic control. We can voluntarily control it as well, but that's one of the very few things that really is activated both ways. This autonomic nervous system is also very important for us because as we see, it breaks down into the sympathetic and parasympathetic systems. So the sympathetic nervous system then arouses the body, prepares it for action, such as we might need if we're faced with a physical threat like an attack by a tiger, gets us ready to flee or to fight against that threat. Parasympathetic system, on the other hand, calms the body, relaxes it, and promotes rest and recuperation, digestion, replenishing our energy. So we'll get into those systems and how we might hack into those systems for our benefit later on. When we're looking at the neurons in the nervous system, there are some very basic types that we can identify. Sensory neurons. These carry sensory information from our sense receptors and our eyes, ears, skin, and so forth back to our central nervous system. Motor neurons, you probably guessed it, have to do with movement. Motors move things. And so these are those that activates our muscles and glands. And we also then have interneurons, those that connect two or more neurons together. Within the brain itself, the majority of neurons there are going to be interneurons. So our central nervous system then is made up of many neural networks. These neural networks then form as neurons that fire together, wire together, as they say. Okay, so when neurons are activated together, then they become more sensitive to the others in that neural network, and eventually, as we stimulate one, that network is stimulated as well. Notice that neural networks are extremely complex, so we can process information in extremely complex ways. In this illustration, notice all of the different connections that you have with just about a dozen neurons. Now consider that many neural networks have thousands and thousands of neurons. Imagine the complexity of the processing. In fact, in terms of processing complexity, our brain still outstrips even the most advanced computers even today. It's not faster than the computers, but can definitely engage in more complex processing. Now, these neural networks also have the capability to modify with growth and experience. And so when children are very young, when they're babies at first, those neural networks for movement aren't completely formed yet. And so as the child's waving their arms and legs around, as they find that some particular movement is pleasing, those neurons involved with that movement will begin to strengthen their relationships with others until eventually a stronger neural network is formed to the extent that finally the child can voluntarily activate that movement and eventually a movement pattern as they form and boot up these neural networks. Notice these neural networks also can modify with experience. So after they have been set up, if the input changes, well, that neural network can change and we can change our response to that information appropriately. That really is an advantage. And once again, even computers are not able to rewire themselves in that sense, at least not yet. So neural networks, very important. Here's an example of a very simple neural network, and this is involved with what we call the hand withdrawal reflex. So person puts their finger on something that's too hot, receptor cells in their skin detect this and stimulate sensory neurons, which are routed to their spinal cord. Those sensory neurons then activate interneurons. The interneurons, in turn, activate motor neurons that then, traveling down that person's arm, eventually arrive at muscles that they activate causing the muscles to contract and get our hand out of the flame. So this indicates how neural networks, various neurons, work together to accomplish a task, cooperate. Also, if you've noticed, this has happened to you. Maybe you accidentally stuck your finger on the stove. It's pretty hot. The response is instantaneous, right? You say, wow, even before I felt the burn, I had pulled my finger away. How does that work like that? Before I even knew it, I pulled my finger away. Well, notice that, first of all, this whole thing goes on here in your arm and spinal cord. The whole thing doesn't have to first be sent to your brain, processed there, and sent back. So that shortens the distance the information has to travel. And secondly, it's a very simple neural network. There's not a lot of steps it has to go through, so it can occur very quickly. That explains why that phenomenon is kind of weird. Okay, so now let's have a little closer look at the actions of the autonomic nervous system. And first of all, a little more in depth about what goes on when the sympathetic nervous system is activated. Again, this might occur under strong emotions or in cases where we might feel that we are under threat. And in this case, may also activate what we call a fight or flight response. So in this case, what we have is we have the activation of this system. Pupils will dilate so we can see the threat better. Heart begins pumping more rapidly so we can send more blood to our muscles. Our stomach stops digesting because we don't have to digest anything right now. We just need to use all our energy to escape that threat. The pancreas also releases insulin so that we can actually utilize the glucose in our blood. Liver is releasing glucose so that we have lots of energy in our blood. The adrenal gland is releasing epinephrine and norepinephrine to make us more alert, to speed up our heart and bodily processes. And if that particular threat is intense enough, the bladder may relax, and then we're going to have to go and change our underwear afterwards. So in just a moment, I'll have to pause the thing. It looks like I've got some kind of important call here. Hang on. Okay, now let's have a look at the other side of that. The parasympathetic nervous system, when we feel that we are not in danger, then this system calms the body, allowing us to rest and digest. So you might see much of it seems to be kind of reversing, going in the opposite direction of those previous processes. Okay, so now we're going to shift to having a look at the influence of the endocrine system on our psychology. The endocrine system is our system of glands which produce hormones. So glands being tissues that produce hormones, chemical substances that travel through the bloodstream to influence areas of the body remote from where that gland is. You might say that our hormones then are our body's slower chemical communication system. The neurotransmitters are the rapid communication system. What do we have? We have numerous ones that we're going to be looking at. Don't worry if you haven't written all this down. We'll take one at a time. Okay. So we'll be looking particularly at the pituitary gland. We'll be looking at the thyroid gland, adrenal glands, pancreas, and then the sexual glands, testes, and ovaries. So hormones are chemicals made by endocrine glands secreted into the bloodstream, affecting the brain and many other tissues of the body. For example, epinephrine, which we also know as adrenaline, released by the adrenal glands just atop our kidneys, increases heart rate, blood pressure, blood sugar, and feelings of arousal during emergency threat kind of situations. The pituitary gland is an important one for us psychologically. Often it's called the master gland because it regulates numerous other glands. Here are some examples. It's part of the HPA axis, as it's called. H stands for hypothalamus, a brain structure that is right above the pituitary, which is right here at the base of the brain. The pituitary receiving signals from the hypothalamus then, when we're facing a threat, sends a hormone to the adrenal glands that then causes them to release that epinephrine, that adrenaline. So HPA axis when we're under threat. The pituitary gland is also the source of oxytocin. Now, oxytocin has some important psychological functions. They find that it is involved with bonding with others, forming close relationships where we have a close attachment with others. It's also released when we engage in pleasurable interactions with other people. And so social interaction and the pleasure that we might get as a part of that is related to this as well. Okay. Also, the pituitary gland has a role in the sexual response cycle. So this again involves the hypothalamus, that brain structure. If we encounter something that is sexually arousing, then the hypothalamus releases a substance that stimulates the pituitary that then in turn stimulates our sexual glands to release hormones that increase that level of arousal. So it's kind of as if there's a loop, hypothalamus, pituitary, sexual glands, and those hormones then coming back and having the effect of increasing that arousal. The thyroid gland also may have an influence on our psychology. The thyroid gland, found in the neck area, regulates our metabolic rate, how fast various chemical reactions occur in our body. If the thyroid malfunctions, we may become depressed as a result. So if someone goes to the doctor and they say, "Doc, I've been depressed," and there are any signs of thyroid malfunctioning, a well-informed doctor is going to test for thyroid function before sitting down and prescribing an antidepressant. Indeed, all they may need to do is correct that thyroid malfunction. Next, we have on our outline the adrenal glands producing epinephrine, norepinephrine, released in situations where we are under threat, and also cortisol, a stress hormone as well, stress hormone. We have the pancreas that's found here near the liver, producing the hormone insulin. Many of you are familiar with that and know it has something to do with regulating our blood sugar. And indeed, if our blood sugar gets out of the right range, too high or too low, we might either go into a coma and lose consciousness, or we might have other problems like edginess, irritability, feeling faint, or simply feeling sluggish, you name it. So pretty important for our psychology as well as our physical health as well. Then we have the sexual glands, the ovaries in females, testes in males. So these then produce hormones involved with sexual reproduction as well as some other functions, some of them psychological. So testosterone we associate most with males because males have several times more of this than females, but females also have a certain amount of testosterone. Normal levels of testosterone are necessary for normal levels of sexual interest. If the amount of testosterone that should be there for you, whether you're a male or female, is not there, you will lack interest in sexual activity. Also, they have found that when people are lacking the normal amounts of testosterone, they may feel fatigued, tired, and unmotivated. Now, they've also associated higher levels of testosterone with greater aggressiveness, and studies have shown that repeatedly. For example, when they looked at prison inmates, they found that prison inmates that had committed violent crimes tended to have higher levels of testosterone than those that were there for nonviolent crimes. However, what you should recognize is this doesn't mean that a person having higher levels of testosterone is necessarily violent. Some people may be violent. However, other people may channel that greater aggression into positive pursuits like adventuring, heroism, and athletics and things like that. So it's not always leading to violence. We next have on our list estrogen, progesterone, and these are produced in much greater levels in females and are involved with regulating the menstrual cycle. One of the interesting things they found out is that as levels of estrogen rise and fall during the menstrual cycle, that levels of serotonin frequently follow that in the brain, which might help account for why some women find that they are more anxious or maybe feel a little bit down at certain points in the menstrual cycle. So that may have some influence on those cyclic mood variations. Now, you also find some estrogen found in males as well. You might say, well, what for? Well, there's other processes in the body it's needed for. For example, they found that estrogen is involved with memory formation as well as plays a small role there. So needed in males as well, just not as much. Okay, so let's next have a look at how have we learned all the things that we know about the brain? What techniques may be used to study it? So one of the oldest techniques is the creation of brain lesions. This involves experimentally destroying brain tissue to study its effect on animal behavior. So notice this is all only done in animal research. So what the researcher does then is in removing that brain tissue or destroying it, they can note changes in the animal's behavior, which would indicate what that part of the brain is doing. Here's an example. This is a brain lesion produced by Hubel back in 1990. This is the visual cortex of a cat. And it's not pleasant to look at and think about. However, what you should know is from Hubel and Wiesel's study of cats' visual cortex in this manner, they learned about these specialized cells called feature detectors, which now has led scientists to actually be able to begin restoring sight to people that have damage to their visual cortex. So ultimately, it's beginning to have a benefit for humans. Still not very pleasant to think about, but nonetheless may yield very important information. Another technique that may be used is clinical observation. So we can observe people who've had various brain disorders that disrupt specific areas of their brain and how that impacts their behavior, their cognitive processes, emotional processes, and so forth. Or we might study changes that have occurred in the brain structure of those who have died that have had neurological or psychiatric diseases. And here you can see brains that have been donated to science that can be studied. We may learn things about various mental functions by studying these people's brains. Then you have the electroencephalogram. This is able to detect electrical waves being produced by the brain. Now, when people look at this, they always like, oh my gosh, that looks creepy. But in fact, know that those electrodes here are simply affixed to the little girl's scalp by using a little gel. They're not, drilled in there or anything like that. They're just held by gel. And so in detecting those electrical waves, this is something that we might be able to detect abnormalities in the way the brain is functioning. Also, in studying these electrical waves, we've been able to identify electrical waves coming from various areas of the brain that are associated with certain types of mental activities, brain waves that are associated with various types of consciousness, like wakefulness, various stages of sleep, and so forth. So, pretty good in helping us understand how the brain is functioning. There's also the PET scan. The PET scan enables us to see where the brain has recently been active. The way it works is they inject a special form of glucose in the person's bloodstream that is slightly radioactive, and sensitive equipment then can detect where the brain is utilizing that glucose. So we can have a person do some sort of mental task and then track what part of their brain was most active as they did so. By the way, you might be concerned about that radioactivity. To what extent is that substance radioactive? I guess about equivalent to getting maybe 6 X-rays. So not terribly threatening, but obviously we would want to limit the use of that in research. One of the biggest advances in studying the brain has been the development of the MRI. The MRI can give us clear pictures of the brain's structure without exposing the person to any radioactivity. And so, in being able to see the brain structure, we may be able to see differences in that way, in the way the brain is structured. And for example, you have here uh, these two MRI scans. The one on the left, normal person, you'll notice that these ventricles, these empty areas, fluid-filled areas, are smaller in the normal person on the left as opposed to the person on the right who happened to have schizophrenia and took psychiatric medications for many years. Now, interesting in this case, it's actually a composite of many people's brain scans in each particular situation. But we're not, from this, we can't tell for sure whether those changes are due to the progression of schizophrenia and what it might do to the brain, or whether those might have something to do with the psychiatric drugs that people had to take as a result. Nonetheless, we can see that there is a difference, enlargement of these ventricles and some reduction in brain tissue. The MRI also can give us more information. The functional MRI, denoted by small f in front of MRI, can also show us brain activity as well as a picture of its structure. So in this picture here, you have a person who is lying about their poker hand. And you can see that this MRI shows particular activity in these areas. And so you may be thinking, wow, so now we can use the MRI as a lie detector. Well, not quite. It's not quite good enough yet, because in fact, there are numerous other activities that activate similar areas of the brain. We're not yet able to quite distinguish between those activities and lying just yet. Maybe someday we will, as the resolution of the MRI improves and we study these things further. Notice also that MRI is superior to PET scan, and not only does it show us the activity going on in the brain, but we can now track changes in the activity down to a few milliseconds, whereas the PET scan, maybe at best 15 minutes. So much more responsive and also much more detailed. And by the way, going back to the PET scan doesn't give us a good picture at all of the brain structure. So it's also has that limitation. So MRI has been really great advance in studying the brain. Okay, so we're going to call that it for this segment of the videos, and we'll continue part 2 with specific brain structures and their functions, and also brain lateralization, what each side of the brain may be specialized for. So we'll get to that. Have a great day.